أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم. My topic of research was epidemiological investigation of foot and mouth disease virus in bovines and companion animals, and my supervisory committee included Professor Dr. Sajadur Rahman and Dr. Muhammad Abu Bakr, Dr. Muhammad Imran Arshad, and Dr. Khurram Ashfaq. Personal profile is that I completed my DVM in 2005 from University of Agriculture, Faisalabad. After that, I completed MSc Honors in 2007 from same university, and then I joined as veterinary officer in Livestock and Dairy Development Department in 2008 and worked there as veterinary officer up to 2019. After that, I got chance and got admission in PhD, and now I'm PhD scholar. A little bit about the overview of livestock senses. Livestock contributes 61.89% to the agriculture and 14.04% to the gross domestic production of Pakistan. Census of livestock in Pakistan is 213.1 million, and highest of that is goat population. Goats are 80.3 millions, followed by cattle 51.5 million, and then buffaloes 42.4 million, and sheep 31.6 millions, and finally camels 1.1 million. Out of 213.1 million, share of district Bahawalpur is 2.22 million. And in district Bahawalpur, population of cattle is highest, that is 0.84 million, followed by the population of goats 0.76 millions. Little bit about the economic impacts of FMD. Due to FMD, world has to face economic losses about 27.5 billions per year, and FMD outbreak cause losses of 1.5 billion per year. Pakistan has to face 692 million losses up till now, and there are two types of impacts caused by FMD: drop in milk production and meat production, abortions and deaths occur due to FMD. Indirect economic losses included loss of drought power, transportation, and cost of implementing FMD control programs. Little bit about foot and mouth disease. This is highly contagious acute disease of all cloven-footed animals, including domestic and wild animals, characterized by high fever up to 104 to 105 degree Fahrenheit, and also characterized by the formation of primary vesicles and secondary vesicles, which we can say them as blisters formation on mouth and hooves area. In mouth, whole buccal cavity is infected, and this disease is caused by the FMD virus of genus Epthovirus of Picorna viridae family. Incubation period of the virus is 1 to 7 days, and there are 7 serotypes, including serotype O, Asia 1 and A, which are more prevalent in Pakistan, and the other 4 they are prevalent in other parts of the country, and they are SAT1, SAT2, SAT3 and serotype C. Epidemiology and worldwide status. Disease is mostly endemic in most of the world, means that there is continuous and regular occurrence of the disease in certain population, in susceptible population, which are the cattle, buffalo, sheep, goats. There is continuous and regular occurrence of the disease in such population. Cases of FMD are 10.74% out of all contagious diseases prevailing in the world. Morbidity of the disease is 100%. The susceptible population that is affected by FMD 100%, and mortality rate is very high in young calves, and case fatality ratio is 15.11%. Nowadays, most of the world is suffering from FMD, excluding USA, Canada, Mexico, Western Europe, Australia, and New Zealand. Transmission of foot and mouth disease. Transmission may occur through direct contact and indirect contact. Through direct contact, breath actually causes the transmission of the virus, and secretions and excretions of the animal are also responsible for the transmission of the disease. When other animals come in contact with the breath of the infected animal and also with secretions and excretions of the infected animal, they are also infected with the disease. Animal products like milk, meat, rest of the carcass, they are also responsible for the indirect transmission of the disease. Now is the pathogenesis of foot and mouth disease, that is caused actually by the inhalation or ingestion of the virus into the stomach. Inhalation of the virus cause entry of the virus into the upper respiratory tract, and ingestion of the virus cause replication of the virus in the alimentary tract. That is followed by viremia, and then pyrexia occurs. After pyrexia, primary vesicles, they are formed on the lips, oral mucosa, and also on the hooves in 1 to 4 days. And after that, the secondary vesicles, they are also formed. Then hydropic degeneration of the vesicles occurs that leads to necrosis, and if not treated, so many losses occurs. In young calves, virus localizes in the myocardium, leading to the tiger heart disease and cause the high rate of the deaths in the young calves. Here are some pics obtained from the field regarding clinical signs of foot and mouth disease. We can see here excessive salivation in the mouth of the cow, ropey excessive salivation, and here we can see sloughing of the epithelium after vesicle formation on hoof area, sloughing of epithelium and vesicle formation on the dental pad area, and here a sloughing of epithelium on the tongue area is very evident. Treatment of foot and mouth disease which we followed at veterinary institutions, they are the topical application of glycerin and boric acid locally on the wound area, that is very effective. And we should also, and we are doing this, washing with antiseptics like potassium permanganate solution, that is very effective. And then dressing to prevent secondary bacterial infection, that is also very necessary. Antipyretics are given like flunixin, meglumine, or meloxicam, and broad spectrum antibiotics including penicillin. So many names like Penbiotic, probiotics, and Penivit, etc. 5 grams. Injections are available and they are applied and they are very effective. Fly repellents, they are also given to avoid the maggot wounds. So now the objectives of our research, there were two main objectives of our research. Number one, to study the seroprevalence of foot and mouth disease virus and its variability in bovines in district Bahawalpur, and number two, to study the seroprevalence and foot and mouth disease virus variability among domesticated animals in district Bahawalpur. To meet those two objectives, we followed this methodology: seroprevalence and molecular identification of foot and mouth disease virus. First of all, sample size was calculated by using this formula: n is equal to 1.9 square into prevalence expected into 1 minus prevalence expected over alpha square or d square. That is actually the absolute Percean required. Seroprevalence and molecular studies. To complete seroprevalence and molecular studies, we made 3 groups. Number 1 group was of vaccinated bovines. Number of samples were 436. Number 2 group was Non-vaccinated bovines, number of samples were 402, and group 3 was containing domesticated animals having 417 number of samples. Area of study was 5 tehsils of district Bahawalpur. 5 tehsils of district Bahawalpur, including Ahmadpur East, Tehsil, Bahawalpur, Yazman, Hasilpur, Khairpur Mewali. So, first of all, seroprevalence 1 study was completed by adopting two techniques. Number 1, NSP ELISA technique, which is actually based upon DVI technique, that is differentiation of infected animals from vaccinated animals, and SPC ELISA technique that was used for the differential identification of FMD virus serotypes. So seroprevalence studies were completed by using these two techniques, and after that molecular identification was completed by taking oral swabs and performing RT-PCR. That is geological map of tehsils distributed in the Bahawalpur district of Punjab, Pakistan. Here we can see this is a map of Pakistan, and in map Punjab, and in Punjab we can see these are the 5 tehsils of district Bahawalpur. Here bigger one is tehsil Yazman, and that is mostly towards northeast area. This tehsil is tehsil Ahmadpur, and after that this one is tehsil Bahawalpur. A smaller one is tehsil Khairpur Mewali, and this one is Hasilpur tehsil. Seroprevalence studies 1. Application of NSP ELISA or 3ABC trapping ELISA test for the differentiation of infected and vaccinated population. Here is scheme of distribution of test and control sera diluted at 1/100 in ELISA buffer in carrier microplate for NSP ELISA. Here is a scheme of distribution in the carrier plate. We can see there are 45 samples diluted over here in carrier plate. Positive control sample, weak positive control sample, and negative control samples were also diluted, and ratio was 1/100. It means that 95 microliter of the ELISA buffer was added in each well, and 5 microliter of the test samples were added up to 45 wells, and 5 microliter of the controls were added in all these wells. After dilution, this was the layout for NSP or 3ABC ELISA, trapping ELISA. 45 samples were applied in alternate columns. First column was containing already 3ABC trapped ELISA, and second column was only containing buffer. Third column and similarly alternate columns were containing 3ABC trapped ELISA, 3ABC trapped antigen. And these columns alternatively they were only containing buffer solution up to 45 wells. Similarly, control samples were also applied in duplicate. In this way, here we can explain that 40 microliter of the test samples were applied in these columns up to 45 wells in duplicate columns, and 10 microliter of the test sample, 45 microliter of the ELISA buffer was added in these alternate columns. After that, in all these columns, and then 10 microliter of the test samples were applied up to 45 wells, and 10 microliter of the controls were applied. After that, incubation was performed for 1 hour at 25 degree centigrade temperature, and after performing that, wells were emptied, and then 200 microliter of the washing solution was added, and incubated for 3 minutes. After 3 minutes, wells were filled with horseradish peroxidase conjugate at the rate of 50 microliter per well. Incubation was performed for 1 hour again at 25 degree centigrade temperature, and then again wells were emptied and washed for 3 times. After washing for 3 times, chromogen or substrate solution was added at the rate of 50 microliter per well, and plate was kept for 25 minutes in the dark after covering the plate. After that, finally, stop solution was added at the rate of 50 microliter per well, and optical density was noted. Net optical density was equal to optical density of the test serum and control serum in well without antigen minus optical density of the wells containing antigen. So after that, percentage positivity that was calculated, and that was equal to the net optical density of the test serum divided by net optical density of positive control serum multiplied by 100. Our test validity criteria was, in this case, in NSP ELISA, that positive control serum gave a net optical density value higher than 1, usually ranging from 1 to 2, and percentage positivity of weak control serum that was more than 10% but less than 50%, and negative control serum was giving less than 10% percentage positivity. So here cutoff value was more than or equal to 10%. So the test samples which were showing more than or equal to 10% percentage positivity, they were regarded as positive, and vice versa. After that, seroprevalence 2 studies were completed by using this application of solid phase competent ELISA for differential identification of foot and mouth disease serotypes prevailing in bovines and other domesticated animals. This is scheme of sera distribution for solid phase competitive ELISA plate. First of all, here at 4 negative controls were applied on position E11, F11, G11 and H11. Here we can see the microplate layout scheme of sera distribution. After application of negative controls at the rate of 50 microliter in these wells, we added 67.5 microliter of the ELISA buffer wa alaikum salam at position G12, and then after and 50 microliter was added at position H12 of ELISA buffer was added here. After that 7.5 microliter of the positive control serum was added over here at position G12, and total volume became 75 microliter and dilution became 1/10. Then we took 25 microliter from here and added that in this well H12 1/30 dilution was obtained, and after that 90 test samples were applied over here at the rate of 5 microliter per well, and 45 microliter ELISA buffer was added in all wells of the microplate. After that, incubation was performed, and at 25 degree centigrade temperature, incubation was performed for 1 hour. After that, without emptying the wells, we added horseradish peroxidase conjugate at the rate of 25 microliter in each well, and then again incubation was performed for 1 hour at 25 degree centigrade temperature, and then after that wells were emptied and washing solution was filled at the rate of 200 microliter in each well, and then incubation was performed for 3 minutes. After that again wells were emptied and After that, again washing solution was added in each well. 3 times washing was performed, and finally chromogen was added, that was actually substrate at the rate of 50 microliter per well, and incubation was performed for 25 minutes. After covering the plate, at the last, stop solution was added at the rate of 50 microliter per well, and then after that reference optical density was calculated by taking the mean optical density of the four negative control wells. After calculating that, percentage inhibition of the positive control sera and test sera was calculated, and that was equal to 100 minus serum optical density divided by reference optical density into 100. Our test validity criteria in this case was percentage inhibition of positive control was more than or equal to 90% at 1/10 dilution, and it was more than 50% at 1/30 dilution. Negative control wells showed more than or equal to 1 optical density, and our cutoff value in this case in solid phase competitive ELISA was more than or equal to 70%. Means the test samples which were showing more than or equal to 70% percentage inhibition, they were regarded as positive, and vice versa. Here are some results of microplates showing solid phase competitive ELISA results. After that, finally, molecular identification of foot and mouth disease virus from bovines and domesticated animals was performed by taking RNA extraction from oral swabs by using GeneJet purification kit. And then cDNA formation was completed by using forward primers having this configuration, and reverse primers using these configurations. Here is there are some components showing the reaction mixture. Total RNA was 5 microliter. Random primers were taken 1 microliter. DNTP's mix was taken 1 microliter, and nucleus free water was 7.5 microliter. All these things were added and heated at 65 degrees centigrade temperature for 5 minutes, incubated on ice for 1 minute, and then centrifuged briefly. After that, RT buffer was added 4 microliter, RNase of ribonuclease 0.5 microliter, and easy script RTase was added 1 microliter. All these things and previous things, all these things and these things, they were briefly centrifuged, incubated for 2 times. First of all, incubated at 25 degrees centigrade for 10 minutes, and then incubated at 42 degrees centigrade for 50 minutes. Reaction was closed at 85 degrees centigrade temperature for 5 minutes and chilled on ice. 1 microliter of E. coli RNase H was added, and lastly incubation was performed at 37 degrees centigrade for 20 minutes. After cDNA formation, here are some conditions for the amplification of FMD virus cDNA. PCR was performed in these 3 steps: denaturation of DNA, annealing, and expansion. Taq polymerase reaction was performed at 94 degrees centigrade temperature for 10 minutes in 1 cycle. Denaturation was performed at 94 degrees centigrade temperature for 1 minute. Annealing at 59 degrees centigrade temperature for 1 minute, and finally expansion of the DNA was completed at 72 degrees centigrade temperature for 1 minute in 35 cycles. After performing that, sequence analysis and statistical analysis was performed after observing bands of the positive samples. So positive samples were submitted to Advanced Bioscience International Lab, Lahore. Phylogenetic tree was constructed and then visualized using MEGA6 program. Phylogenetic tree was constructed by employing BioEdit. Statistical analysis was completed by using Minitab software, and p-value was calculated to determine statistical analysis. P-value showing equal to 0.05 or less than 0.05, they were regarded as statistically significant. Here are the results based on solid phase, based on NSP ELISA. Tehsil-wise distribution of seroprevalence of foot and mouth disease virus in bovine population based on NSP ELISA. And we can discuss all these graphs one by one in next video, inshallah. Thank you very much. Jazakallahu khaira. Allahumma salli ala Muhammadin wa ali Muhammad wa ajil farajahum.